Welcome, everyone. Today, as our guest, we have Jörn Diedrichsen, who graduated completed neuroscience training and statistics at UC Berkeley with Rich Ivory, and then his postdoc with me here at Johns Hopkins. Afterwards, he received a faculty appointment at Bangor University in the UK, and then University College London. And now he is a research chair in computational neuroscience at the University of Western Ontario in Canada. His laboratory has been a leader in using behavioral, computational, and functional imaging methods to study the human brain during both skill acquisition and the connectivity between regions of the brain. Arguably, he's been leading the best lab in the world in understanding the relationship between the cerebellum and the rest of the brain. It's great to have you, Jörn.
Great. Thank you, Reza. It's a pleasure to be back in this really great talk series. So yeah, what I want to do today is kind of give a little bit update on our work on trying to understand the connectivity between the cerebellum and the neocortex in humans. Now, why are we interested in this? So, well, really, we are interested in answering the question, the multi-million-dollar question: what is the function of the human cerebellum, or the cerebellum in general? That's what we actually trying to answer, like many other people. And if we ask this question, what is the function of the cerebellum, we need to keep in mind that that there are many different functional regions in the cerebellum. Each of these functional regions is likely connected to a different set of cortical and subcortical regions. So asking this question is really what I would call a horizontal question that is very difficult, I think, to answer. You know, nobody really in their right mind asks the question, what is the function of the neocortex? And you might as well, by rights, ask the same question. So I think it's useful to maybe start with an easier question, which I would call a vertical question, which is, let's think about what is the function of each of these cortical cerebellar units or loops in the behavior first, and then ask the question, within each of these loops, what does the cerebellum contribute to the processing within this loop? And I think that's a much more tractable problem. Now, if we want to do this, of course, the thing that we need to know is, what are these modules and which ones for each cerebellar regions, what neocortical and subcortical areas does it connect to, and which ones do we need to analyze together? And so basically what we need to do, if we want to do this in humans, we need to map the cortico-cerebellar connectivity in humans. Now, the methods we have are unfortunately limited. So we don't have access to the viral tracing that we have in animals. And the fusion tensor imaging doesn't have the resolution to really resolve the fine grain connectivity through the pontine nuclei and through the dentate nuclei. So this is a problem. So really, the approach that has been dominant in trying to understand this is using functional imaging signals. And this is something that was really spearheaded by, not the first, but the first really convincing application of this was done by Randy Buckner. Now 15 years ago. So Randy collected, together with Thomas Yeo, he collected a large data set of 1,000 subjects of functional magnetic resonance imaging data, while the subjects lie in the scanner and just rested there. Okay, and so you can see that there's some fluctuations in each of these voxels. And what he did then, using this data, is he clustered the neocortex into so-called networks. So regions or vertices that in the neocortex that fluctuated together across these 1,000 subjects, across this time that they were in the scanner. And he clustered them, and he came up with these canonical 17 networks. Randy then took the cerebellar data from these 1,000 subjects and went to each voxel and just correlated the time series observed at the same time at that voxel with these 17 networks, and then just found the network that was best corresponding to it. So this would be network 16 here in this case, and then colored this voxel by that color. Okay, and he came up with this map that actually looks very similar to the one that we produced on task-based imaging, and so it's really a very powerful approach. So I have to admit that up to that point, you know, I kind of grew up with a training. I was trained to that if you do experiments, you need to have an independent variable. You need to have a task if you want to identify a biological system. It's good to excite the system to identify it. So every engineer learns this. And so I was highly critical of resting state up to that point. And it was really when I reviewed this paper from Randy in 2010 that I was like, maybe there is something about resting state. There is something really profound in these fluctuations that we can learn. Okay? So when we started our own work, I wanted to really get to grasp this, understanding the relationship between resting state and task-based fMRI. And so that's in the first part of the talk, that's what I'm going to talk about. And so I apologize for the non-images. It's going to get a little bit technical and maybe a little bit specialized, but I think it's a very important question because there's a lot of practical relevance. And then in the second part of the talk, I'm going to use this to then show you what we've done in building a new connectivity model between cortex and cerebellum. So when I say we, I really mean Caroline Eckenkofen, who just left the lab, and she now has her own group at Oxford. And so she was very interested, together with me, in trying to really get to grasp with what's different between resting, what answers do we get from resting state and from task-based imaging. So in resting state, the idea is the following. You put people in the scanner, and you don't give them anything. You just run the scanner for 20 minutes. And so basically you can think about the state of the brain at rest, that there's some activity maybe in this cerebellar region 1, and also in this cortical region 2 and 3. Of course, if this was stable, then you wouldn't see anything. But what has been shown in the late 90s is that there are these interesting fluctuations in rest, in the neural state, in the fMRI signal, in the entire brain. And that the structure of these fluctuations reveals something about the underlying connectivity, the underlying anatomical connectivity. So for example, here, region 1 might co-vary with region 3, and so that could be taken as evidence that these two regions are connected. Now, since then, resting state imaging really has taken the field by storm. Okay, and there are many reasons for this. One, it's very easy to implement. You can do this in neonates, you can do it in people that are impaired. You just put them in the scanner and you scan. So it's very easy to acquire. And at this point, there's a very, very large data set, population data set on resting state data available. You can download reams of resting state data from the web and analyze it for many different types of population. So from a clinical relevance and a kind of population health relevance, this is really important data, okay, that we really need to get to grasp with and understand. So this is why we just can't dismiss it outright. Now, what I didn't like about it always was that it's hard to separate, hard to know whether these fluctuations are real neural fluctuations, so really like reveal something about the brain, or they're just artifacts of the scanning, like physiological and scanning physical artifacts in the signal. And if you have done fMRI, you know there are a lot of artifacts and noise sources on this signal. It's a very noisy signal. Okay, so and basically, you have very little control. Now, if you do task-based imaging, you have the control. And what you do is basically, and this is like, basically what I've been taught, you put somebody at rest, then at some random point, you give them a task, and you drive the brain state to this new state, and then it returns, and you can do this repeatedly. And that gives you a leverage, an independent variable, where you can separate out what is... Hello?
Thanks so much.
That's ask questions if you want in between. Anyway, so this gives you a handle to actually separate out what you can, what is task induced and what is measurement noise of random fluctuations. Okay, so you have this handle. Now the problem is, of course, if you want to use this data to build connectivity models, so look at correlations between regions, you're going to be dependent on the task that you choose. So for example, if you choose this task, region 1, the cerebellar region, might co-vary with cortical region 2. You might pick another task where the state is here and region 1 side might suddenly correlate with region 3. So it will be dependent on the task set. Okay, and of course it's harder to implement. Now, when we started, we thought like, what can we do? Can we create a hybrid approach? And so we call this approach now the multitask or active state connectivity approach. Okay, and so the theoretical step that we kind of really think is about not to think about tasks that we inherently as something that we're inherently interested in, but think about task as a random factor. So think about the brain, what you do in everyday life, right? And you go about and you do all kinds of things. You listen to talks, you go and have lunch, you talk to somebody, you watch a movie, you sit on the sofa and rest, and so on. During your waking waking life, you engage in all kinds of different tasks. And, basically the idea is like, if we could sample those activities, okay, and put them in the scanner, we would really have basically a picture of what the brain does in the active state in normal life. Okay, so that's kind of the idea. So the idea is to sample many different tasks, and it doesn't really matter which tasks they are, ultimately, as long as they're broad and cover the spectrum of mental activity broadly, it's good. And then you can drive the brain through all these states actively. And the idea or the hope is that ultimately, because it's a random fact and because you sample, the larger your sample gets and the more often you sample, you're going to converge to the underlying truth, the underlying real connectivity that the brain has. Okay? So it's kind of a natural statistics approach to stars. Now, we don't have a good natural statistics of mental activity, unfortunately, but basically what we did is we just looked in the cognitive neuroscience literature and just sampled tasks at random and implemented them. Now, the advantage, of course, that you still have an experimental control, so you can drive the brain actively through these different states, so you don't give up, like in resting state, you don't give up on that handle. The problem, of course, when we started is that nobody had done that before, so we needed to go back and really do this. And so this is what started in 2015 in my lab when Maeve King was a graduate student here. And so she did this task where she took many subjects, or 24 subjects, and she gave them many different tasks, about 50 different task conditions, and scanned them. And so basically what we get is that what we can do in the cerebellum, we can elicit these very reliable activity patterns in each of these tasks that actually vary quite widely. And we've used this in many applications to really study the functional organization of the cerebellum. Now, what we wanted to do is really to understand what is the difference to resting state. And luckily, and we hadn't used this data up to that point, we also acquired 20 minutes of resting state data from those same subjects on the same scanner, usually either in the same session or in a slightly different session, but very comparable functional imaging data with the same sequence. And so what we can do now, we can build connectivity models, or looking at answers we get from the resting state data, and we can compare that to different ways of putting together a 20-minute task scan from different snippets of these 2 hours that we have, okay, from those subjects. Okay, and so we are, of course, interested in connectivity modeling, but we also, the lab has also done parcellation. And it turns out if you want a general answer about the differences between task-based and resting state, you can look one step before building these models. You can actually look at what's called the sufficient statistics. So the criteria or the characteristics of the data is really important for this. And it turns out that for all of these brain models, it's really this voxel or vertex by vertex covariance matrix of the data that is the critical statistics. So in our case, it looks like this, right? So we have, if we take some either resting or active state data, we get a correlation or covariance between different cortical regions. Okay, so this would be important if you wanted to build a cortical parcellation like Thomas Yeo did. If you want to build a cerebellar parcellation, then this covariance matrix, the cerebellum to cerebellum covariance matrix, becomes important. And if you're interested in cortical-cerebellar connectivity models, then it's the cortical-cerebellar covariance matrix, that part that is important. Okay? Now, these different covariance matrices, you can look now at these covariance matrices themselves. And of course, they might be different between rest and different tasks. Okay, but ultimately, what we want to do, or what we assume, is that they all reflect some underlying biological constraints. Okay, so they have something in common. It has been shown that this is the case. Okay, and so we want to learn actually in this, biological truth in a way. Okay, but what we have is, of course, fMRI data that is full of measurement noise. So we can now take rest data or different tasks, and we now measure them, and we get some data. Now, people know that there's measurement data. People aren't stupid. And so nobody really uses a raw time series for resting state data. So that over the time, as the approach has matured, people have developed cleaning algorithms that are supposed to reduce measurement noise. So for example, there's the state of the art is really an independent component analysis on the fMRI data, and then identifying and throwing out the components that are not, that look like noise, measurement noise. And another approach on top of this is that you then go and upweight or give higher weight to the time series that look like other time series in the brain. Okay, so you look at biologically signals that are biologically reasonable, and this is called connectivity fingerprinting. Okay, The typical approach in active state or in task-based data is the one that I learned when I was a graduate student, namely running a first-order linear model. So you basically take the time series and then you estimate from maybe these 300 time points that you have measured, 300 seconds of a run, you estimate for the 17 tasks that you have in your run, you estimate 17 values or contrast maps. That tells you what the average activity during this task is. And those you can look at how well you can replicate them across runs. Okay. Of course, you can also take the task-based data and treat it like resting state data, right? You just forget about the task and just treat it like a time series. You can do that as well. Okay, good. So we wanted to compare these different approaches. And of course, we don't know what the biological truth is. But the idea is that if we had a new task set on the same subjects, okay, a completely different task and remeasure the connectivity or covariance matrix, we want to get close to this answer. Okay, and luckily in this data set, in this large data set, we had another 2 hours of the same subject with the same scanner on a completely different set of tasks. Okay, so and we use this as our target. Okay, let's see what we get. So first of all, the question was, how dependent is the answer really on the task that we're choosing? And so what we did here, or what Caro did, was to estimate covariance matrix from artificially put together fMRI data, 20 minutes of fMRI data that contain only the activity during 2 tasks. Okay, so we pick random 2 tasks, maybe rest and some task, and theory of mind and movie watching, and you can see, and these are all relying on different data, independent data, and basically what we then calculated, we calculated the covariance matrix and then looked at how similar these covariance matrices are together. So if 2 dots are very close together, they gave very similar estimates. If they're very far apart, they're systematically different. Okay, so this is multidimensional scaling plot. Okay, so you see that when you select 2 tasks, 20 minutes of 2 tasks, you get a really large spread. Now we can do this for 3 and 6 tasks. You can see that you get already a little bit closer. And so when you select every time the same 17 tasks, but here now, these are different parts of the runs. So you basically, this is still independent data, but it's always the same 17 tasks selected. You get actually a very tight answer. The covariance matrix really homes in onto this middle spot. Okay. So that's good, but we don't know. You might say, well, maybe it depends on these particular 17 tasks that you choose. So now we can look at these other 2 hours that we have from the same subjects on completely other 17 tasks that are completely different. And what we find is that that is actually very close to that covariance matrix. So it seems like when you have 2 tasks, yes, it's a problem. You're really task dependent. You really, the covariance matrix you get depends on the task that you choose. The more tasks you sample, the closer you get to this central point that you also get with a completely different task set. Okay, so that's good. So now the question we had is like, when we took, this is 20 minutes of task data, well, how would rest fall? And I think if you asked Randy Buckner, he would bet that rest would fall right smack here in the middle of the bullseye, close what he calls the biological truth, okay? Now, that's not what we found. We found actually that rest was substantially different. And to appreciate this difference, we need to actually plot the data a little bit differently. So what you saw here is that a sleight of hand, I plotted only dimension 2 and dimension 3 of the multidimensional scaling plot. And it turns out that dimension 1 is actually the important dimension to distinguish resting state from task-based data. So you kind of need to take this cloud and now rotate this 90 degrees, if you follow me. Okay, so that looks like this. And so here, these are the new tasks, here the 17 tasks. And when we look at this dimension, we see that rest is actually falling quite away from that point. Okay, whoops. So here's resting state raw, and it's very far away from the new tasks. Okay, much further away. And as you increase the cleaning, and connectivity fingerprinting, you actually come closer to this answer, but you never quite reach it as well as another task set where you just do a GLM approach. Okay. Now you can also take, of course, the time series from the active state and just treat it like resting state time series. And what you see is the same thing. As you increase the cleaning, you get closer to this underlying biological truth, the real connectivity, but you never quite reach it. Okay. So, what is the reason? What is the systematic differences between a resting state and task-based? And what is this dimension 1? And it's very hard to answer, actually. So like really people that really believe in resting state and really think this is the Nice Bees, they would say, well, resting state can reveal something that task-based imaging, contrast-based imaging cannot reveal at all. Okay, so it's some new magical thing. Now, that might be true, and it's very hard to argue with this, but we think there's good reasons to believe that at least part of that difference is explained by measurement noise. And so one place where we have pretty good guesses what measurement noise looks like and what the biological truth is the connectivity between the superior cerebellum, so this is kind of this blue area here, just the upper rim, and the directly abutting inferior visual and temporal cortices. Okay, because we know that these are separated by a hard membrane called the tentorium, so there's no connections to that. And we know that, like in the monkey, these regions are actually connected to the superior part of the brain and not really to these inferior regions. So we think that at least the connectivity should be stronger to those regions than to these abutting regions. On the other, measurement noise, if it's spatially smooth, meaning like it's induced in different parts, and many different processes in measurement noise would do that, not all, but many, then this correlation might be actually strong. Okay? So what we can do is we just calculate the correlation of these blue voxels with the rest of the brain and these blue voxels with the red voxels and subtract them from each other. So negative value means that this is a stronger correlation than these long-range correlations. And like if you take both task-based or resting state data, you see it's susceptible to this measurement noise. Connectivity fingerprinting is really a good trick to get rid of it and really at least break even. When you take task-based GLM, so you just take these contrast weights, you actually get a positive correlation and see what we would expect that the correlations, the long-range correlations are longer than the short-range correlations. Okay. So to summarize then, to really wrap this up, in comparison active state and resting state, what we found is that like as you increase the number of tasks, maybe like 10 tasks randomly sampled over mental activities, the answers you get in connectivity are increasingly becoming task set independent. Okay. The other thing we find is that there's systematic differences from answers you get from resting state and task-based, and that part of those differences might be due to measurement noise. And that if you want to predict the covariance in a totally new set of tasks, you are better off with using a random sample of other 17 tasks rather than resting state data. Okay. So I apologize. So this is a pretty long-winded introduction on imaging, on active state and resting state, and you might not be interested in. So you might be sitting there and saying, well, what does it tell us about connectivity? Now let me just tell you what we did on trying to understand connectivity. So this kind of started again with Maeve when she was in the lab. So what we did, we took our task maps from the 29 task in the cortex, and we used the cerebellar contrast maps from the same subjects acquired at the same time. And we started to build linear models of trying to predict the activity of each voxel in the cerebellum as a linear combination of all those cortical parcels. Okay, and that gives us a set of connectivity weights. Obviously, this is an ill-posed problem because we usually have less tasks than cortical parcels. Okay, so we need to restrict it somehow. And so we tried different ways of building these models. One is a one-to-one connectivity. So each voxel just connects to one cortical area. So this is like basically what Randy did in his paper. Or we have like some sparse or more distributed connectivity, convergence of cortical areas on these cerebellar regions. And so now we can test these different models in their ability to predict on the now new data set on the same subjects with completely different tasks. We can take the cortical activity, okay, and predict with those weights learned from this data set, we can predict the cerebellar activity pattern in those subjects. Okay, and then we can, of course, measure these and we can compare the two, and then the correlation between the predicted and the observed is a evaluation of how good this model generalizes to new tasks. Okay. And what we found is that there is evidence for connectivity. So the one-to-one is not really a good model. We also found that this convergence of multiple cortical areas might be more pronounced in these cognitive areas of the cerebellum rather than the hand motor areas. And we've replicated these results recently on more data. Okay. But back to our resting state versus task-based, we wanted to know now, okay, what we learned about resting state and task-based, does it generalize to what we found does it generalize really to connectivity models? So what we did was we took 20 minutes of task-based data. So we used the 2 runs of 17 tasks, so 2 times 17 values to estimate a connectivity model. And that gives us a predictive accuracy of this. And then we used 20 minutes of resting state data. So this is 600 data points, and we get this one. So you see that we're actually much better at predicting it from these 17 other tasks. So that kind of replicates what we found in the covariance matrices. Interestingly, if we take the time series from the task-based data, again 20 minutes, what we see is that we get substantially better prediction than the resting state, even though we treated the data exactly the same way. Okay. So we forgot about which tasks are, and we just analyzed it the same way. So that means like just driving the brain through this active state excites the system in a way that lets you identify this connectivity better. So that's kind of good. Now, at the same time, these predictive accuracies aren't quite as good as the task GLM. And this comes back to a review on our paper saying like, well, you do this task GLM, you really need to take the time series. You're going to get much better prediction. It turns out, no, you don't. Okay, so and the reason I think is that there might be more information in the time series, so there might be these fluctuations, but at the same time, you also have measurement noise. Okay, and it's a balance between that probably breaks even or just breaks less than even in this case. Okay, good. But now we have experimental control. We have the task, so we can actually maybe get rid of the measurement noise completely. And the idea is the following. So basically, normally, we take the activity patterns, the 17 task of run 1 in the cerebellum, and predicted by the 17 task measured in the neocortex at the same time. So these two, this run shares, of course, the same measurement noise. Any fluctuations or so on will impact the run 1 and might impact the connectivity. Okay, but we have the same task in run 2. So what we can do is now do a crossed approach that we predict the activity patterns in run 1 from the cortical activity patterns in run 2 and vice versa, and then do that. Okay, and it turns out that gives you actually then on the new task data set a better predictive accuracy. So that really what held us back here was measurement noise that was still there, even though we first of all condensed the data down to 17 data points. Okay, good. All right, great. So that means that if you want to build good connectivity models, what we learned, it's really good to use an active state approach or like drive the system through some states. And at the same time, really use this crossed approach, use the experimental control that you have to really get rid and control measurement noise in a strong way. And that's what we're doing standard-wise in all the connectivity models that I'm going to show you. Okay, so the last 10 minutes of the talk, I'm going to use to show now what the newest application of this to build a good overall connectivity model, because not always do you have data from individual subjects. So there's some value of having a group population-based model of what normally the connectivity is. Okay. Now, when you build a group model, you have, of course, the option of using much more data. You can use many more subjects and many more task conditions. And so this was kind of Maeve's data set here with 24 subjects and 47 task conditions. And when the pandemic hit, we started collecting other data sets that also had subjects, but also then studied them in many different task conditions, and at the same time fully covered the cerebellum, which isn't that many data sets. So we started collecting these, and these are partly from our lab and partly from various collaborators that we got data from. Okay, and so it was really a big group effort to bring all these data sets into a common analysis framework. And so we really have this at the tip of our fingers now, and we call this framework functional fusion. And we're very happy to share. If you have specific questions and you want to test it on these 9 data sets across things, you can contact us, and the code is all available. And we have made many of these pre-processed data sets available. Okay, so I think that's a really good resource for population-based imaging. So we have 200 subjects now and 450 task conditions, roughly, and we can really build these strong models now. Okay, so what I'm going to show you is really the work from Ali Shabazi. So he's an Iranian student who was supposed to come as a master's student to the lab. Unfortunately, he didn't get his student permit, so he's still in Iran. So if anybody of our European listeners wants to offer him a PhD position, he's still up for grasp, and he would be delighted if you contacted him. So we're still waiting for him to come, but he has been working remotely and has been absolutely fantastic on this. So what I'm presenting now for the last 10 minutes is purely his work. So basically what Ali did, he took these different data sets and he trained connectivity models on these different data sets, and then looked at how well they generalize to predict the other data sets. Now, the diagonal, we should say, he always left the subject out from the training. He left the subject out that he's trying to predict. So this is cross-validated. But what you see here is that the diagonal is, of course, higher. These predictive accuracies are higher than the off-diagonal, meaning there is still some data set specificity. Okay, but you can see that these numbers these predictive accuracies are actually pretty high. Now, I should say that these correlations are expressed relative to a group noise ceiling. So these are evaluated on individual subjects and then made relative. 100% would be all the variance that is systematic across subjects. Okay, and we can estimate this by just taking the activity pattern for that task in the cerebellum from all the other subjects and see what's in common, because that's the best thing that you could possibly predict with a group model. Okay, good. Great. So you can see that this is pretty good, except maybe for data sets like the somatotopic data set that are very narrow. So the somatotopic data set is just different bodily movements. Okay, so it's a very narrow domain, and it doesn't generalize well to predict working memory and language and so on. But when you look at these broader data sets, so for example the IBC, it predicts actually quite well, also on the left out data of the HCP task data set. Okay, good. We can, what we then did was just average these connectivity models and we get even better prediction. But Ali realized that for integration, it's actually best to build a mega subject, so a mega group subject where you concatenate these 450 task conditions and that's re-estimated. And that leads to much higher accuracies. So right now, and this is all cross-validated. So every time we leave the entire data set out, and you can see that we predict more than 50% of the explainable variance across all these different tasks with this connectivity model. Now, I should say that what I'm going to show you now is another way of restricting the model, and that is to make the weights non-negative. So we don't allow inhibitory weights from the cortex to the cerebellum with the idea that mossy fiber input is excitatory. And so that leads to very interpretable weights. And the performance hit you take compared to a rich regression model where you allow them to be positive or negative is actually very, very small. Remember that this is a very strong constraint on the model to only have positive weights. Okay, so now we have basically what we can do is we can take a cortical activity pattern on some task. We get the predicted cerebellar activity pattern, and the observed one is very, very close now. Okay. All right, good. So now we can look at how do these weights look like? And to the largest degree, this connectivity model gives really biologically plausible answers and very reasonable things. So for example, if you look into the left motor hand area in the cerebellum, here in lobulus 5, okay? The weights, these are now the weights averaged for these voxels in the cerebellum, and you see the right hemisphere, the right hand area, motor and sensory cortex. Okay, if you take the left, the left cerebellum, if you take the right cerebellar area, you get the left hand motor area. Okay, so very focal, as we showed before. If you take this spatial working memory network here, really medial cruise 1 and cruise 2, you get the mostly the contralateral, middle frontal, intraparietal network here. Okay, and some of this temporal one. If you take this cruise 1, cruise 2 lateral region, there's kind of a default mode network region, you get the anterior temporal cortex, you get middle temporal cortex and temporal parietal junction. Again, more on the contralateral than on the ipsilateral side. Okay, so very reasonable answers. The only surprise really that we had when we looked at this was the orbitofrontal cortex. So you can see here for this resting state network, the weights from the orbitofrontal cortex were very high. And you can also see these orbitofrontal weights here in the other task a little bit. I mean, much less, but they're still there. Okay, so that was really, really puzzling to us. Okay. So then we wanted to know, well, where do these cortical areas project to? So what we can do is we can look at different neocortical areas and look at our connectivity model and just make a map of where in the cerebellum they go to, or where they have weights in the connectivity model. Because they're positive, we can just take the average, so that's very nice. Oh no, sorry. Before I go there, we can we can look at the cortex and ask like which areas of the cortex project more to the cerebellum. So we sum average over the whole cerebellum, and we ask the question, where do these neocortical, where in the neocortex do these things across the cerebellum come from? So what we find is that the parietal cortex and the motor cortex really project a lot to the cerebellum compared to other regions. It's pretty diffuse, so there's connectivity from nearly the entire neocortex. There are a couple of hotspots like the pre-SMA here and the superior temporal cortex that seem to play a big role. And then you have this orbitofrontal cortex, okay, because it really shows up in many different tasks. We can now analyze this by breaking this up into these networks. And what we see is that the sum of the weights, so that just summing up the weights in this cortical regions that project to the cerebellum, you see that that's related, of course, to the size of the region of the neocortex. Okay, so the bigger the region, the more weights they have, and the lines here, the regions that are above the line, really, there's these control regions, so 12 and 13. So these networks here and this sensory motor area, they really punch above their size. So they seem to have especially high density of connections. But the one that really sticks out is limbic B, that's network number 10, that's the orbitofrontal cortex. Okay, so that shows that again. Okay, where do these projections go to? So now I can come to that. So we can look at each network where they go to. We see mostly, again, the expected things. So the visual areas, primary visual cortex projects little, but then the other higher order visual areas really go to the oculomotor vermis, very clear. Hand and foot area, mouth area of motor cortex, you see these different dorsal and ventral attention networks. These are really working memory areas. They go to the working memory areas in the cerebellum. You get these control networks that are very important, that give these again, multi-domain and social linguistic networks. And you get the typical default mode network. So these are networks that are active during rest, especially, or when you follow a narrative or watch a movie or so. The question was this orbitofrontal cortex, how would that look like? Okay, and then when we look at this, so this is that orbitofrontal region. First of all, again, we should see that it's very bright. So there's a lot of connections in the connectivity model to this area. And it's really these cruise 1, cruise 2, really lateral regions. And it's also quite diffuse. Okay, so what the hell is going on? And I still don't have the answer yet. So this is kind of a serendipitous discovery. And the first intuition, of course, is this is still a measurement artifact. Okay, so obviously, whoever has done imaging of the orbitofrontal cortex knows that it's very troublesome regions full of artifacts. So there's a lot of eye movements, head movements, and so on, cardiac cycle. All of those factors will influence this, and maybe also the cerebellum. So maybe we see covariance in the measurement noise. Now, those are not relevant. Those we have ruled out by the fact that our model is crossed. So we predict the activity patterns in run 2 from the cortical activity patterns in run 1. So whatever shared measurement noise is that isn't related to the task is not relevant here. So we can strongly exclude this. Now, it might still be that there are systematic changes in heart rate with the task. So sometimes in tasks, the heart rate goes up together, and that might influence both of them. I start to think that this explanation is unlikely to really explain maybe some of the effect, but maybe not the full effect. Okay, and so Ali and I are still working on this to figure this out. Okay. Now, if at least partly this is true, what does that mean? So that means that the orbitofrontal cortex might really project to these phylogenetically newer areas in the human cerebellum. And because it's relatively diffuse and comes up in many different areas, it could be a measurement artifact, but it could also be that there is a general neuromodulatory input that regulates cerebellar activity, cerebellar input or cerebellar activity that also shows up in the orbitofrontal cortex. So some neuromodulatory system that we haven't discovered yet. Okay. It could be that maybe what we're seeing is climbing fiber activity. Maybe when we get these surprising signals, that induces climbing fiber activity in these areas. And that's what we see here in the orbitofrontal cortex to some degree. We don't know. So this is really the first steps of this. I'm just putting this out there. If anybody has ideas, I'm happy. We would be very pleased to discuss, see if we can find something like this in the animal. Okay. But I want to end with just defending myself. I've been boring you now for nearly 38 minutes about fMRI and connectivity models and so on. And we've been working on this for 5 years. And you might ask, like, what the hell? Why are you so obsessed with it? And I just want to make the case why I think that this model is so important, so useful. Okay, and we have been starting to use earlier versions of this model already. So the first thing is, like, if you do imaging in the cerebellum or recording in the cerebellum, okay, and you discover something about a specific cerebellar functional area, really to understand what's going on there, you need to understand what are the regions, how does it differ or is the same as regions that it talks to. Okay, so you need to see that in the relationship. Otherwise, you're not going to figure out what the role of the cerebellum in these cortico-cerebellar loops is. And for that, you need to know which regions it talks to. And so the connectivity model, you can just average the weights in this region, and immediately you get the set of cortical regions that you need to look at in some large population database. Okay, so that's really useful, and we're using this a lot for that. Now, the other thing that you can do is you can now, if you have a new task, you don't know how much it involves the cerebellum. You can use a connectivity model to just predict the activity pattern across the entire cerebellum. And as I showed you, these predictions are very good. Okay. And then the interesting bit is now when the actual data that you measure violates this. So when there's more activity than predicted by this just simple static connectivity. Okay, and that's really interesting cases where maybe the cerebellum is especially important for those tasks. And this is where we call this selective recruitment. And Laran Shashahani had a nice paper last year showing that you can identify parts of specific conditions in working memory tasks, like encoding 6 items, that seem to engage these working memory areas of the cerebellum more than what would be predicted from the connectivity model. Okay. So that's a really good way of spotting unusual cerebellar activity. Now, the last example is to go into clinical domains. And this is very new and based on, comes from Anila De Mello's lab at UT Southwestern. And so she has a data set from F. Fedorenko in Boston. And obviously that concerns language because that's what F is doing. And so here, first of all, they have this localizer where they play people's sentences or non-words, and then they take the contrast. So this is kind of the language localizer. And what you see then, if you do take this, you observe this language area and a couple of other areas that have been recently described. So this is the region that we call in our atlas S1, okay? That really lights up in the right side of the cerebellum, connecting to the left hemisphere. And our connectivity model, just based on only on the cortical activity, predicts this very well to like, I think, a very pleasing degree of accuracy. Okay. Now, the interesting thing is now they also have a patient who has a very early perinatal stroke in the left language region. And so in the neocortex, this person, because he doesn't have the left language regions, has reorganized language to the right hemisphere. Okay, so we can measure this. And in the cerebellum, what this patient shows is that suddenly the left language area, the left S1, has completely taken over this language, or mostly taken over this language function. Okay, and so the key question now is this cerebellar reorganization that we see, or this change in activity pattern, is this just a passive consequence of that the neocortex has reorganized? And connectivity is exactly the same. And now, because the right hemisphere does now language, it's the left S1 in the cerebellum that does it? Or is there something compensatory in the cerebellar cortical communication? Okay, and what we can do now is we can take our connectivity model and take the patient's data, cortical data, and now predict what the cerebellar activity should look like if the connectivity was the same. And so now we can compare the two, and make inferences about whether these additional activations on the right side or compared to other tasks are really unusual or not. And I won't steal the thunder. I'll let Anila, when she gives a talk in this symposium, give you the answer on this in the update. Okay, great. So I want to end with just acknowledging all the people, especially Caroline Nettenkopf and Ali Shabazi, who have done the most recent work, but overall the whole team in the lab, our collaborators and the funding sources. And I thank you for your attention.
It was marvelous, Jörn. Thank you so much. Please go ahead and raise your hand if you have a question. I'll be happy to call on you. Jörn, the comparison between resting state and the active task, to what extent imagining doing that task would produce something similar?
Just imagining doing the task? Ooh,
yeah.
I mean, some tasks have imagination in them. So, for example, there is a task that we use to get these really lateral areas, or these Broadman area 10 is like, imagine playing your like motor imagery, right? So imagine playing playing a game of tennis, for example. And you get some activity. It's never quite as strong as the real thing. So actually moving, actually solving a puzzle or something gives you much stronger activity. So you can get. I mean, it's part of what we do in everyday life. We imagine things, right? That's what the brain does. But we also do a lot of active stuff. So in the sense of, so we usually rely on very active tasks. But I think, I mean, it's yeah. I'll leave
Okay,
it there.
all
Yeah.
right. Let's go to Stanford and Jennifer Raymond's lab.
Hi, Jörn. Can you hear
Hey.
me?
Oh yeah, I can hear you fine. Hello.
Hey, that was really fun. You got me thinking, and I have a bunch of questions. Let me ask the very general one, and if there's time later, please come back to me, Reza, for the specific one. But I'm trying to think about the disconnect between the resting state and the active map, and the idea of the connectivity. The map or the. So I mean, the fact that they're different, the anatomy and the connections aren't changing. But a voxel is not like a single thing. It actually is composed of a bunch of different neurons. So I was wondering, like, has anyone tried to understand the disconnect in terms of what kinds of activity patterns, when you put like a little network in each voxel, would yield the same differences that you see when you do the fMRI?
Yeah, I mean, let me understand the question, right? So the first thing is that I don't necessarily think that there is a answer, a covariant, like a true underlying functional connectivity, right? There are biological constraints, like the axons and the synapses don't change at the time scale that we can do this task switching, or that the brain drives through different activities. So the anatomical substrate stays the same. But of course, connectivity is task dependent, the covariance. Now, I should say that is the glass half empty, that it changes. Now, the glass half full, and I kind of didn't emphasize this enough, I think, it is absolutely true that you can take resting state data and way above chance predict what the co-activation in the task space are. So don't get me wrong, like these differences that we see between resting state and task based, right? It's much better. The resting state is much closer to the task based zone than not knowing anything. So you can predict at the individual level from resting state, you can predict task-based activity, okay? And many papers have shown this, including ours, okay? So this is kind of the glass half full. So there is a biological underlying thing that is also individual specific that we can identify. Now, these differences, so again, as I said, there are differences, and we would expect that this covariance matrix really looks different across different tasks and depending on the task. So there's task specificity. And then also, like, maybe rest is something special. The brain is in this idling mode, and how regions talk to each other, we would expect on a network level is different than how they talk to each other when we do tasks. And that's fine, right? I mean, we're not debating.
Can I like throw out two
Yes.
concrete models of what that difference could be? So one possibility is that voxel A in task 1 has like a different set of projection neurons active.
Yeah.
Right. So, or is it that all of the neurons are activated when that, or most when that region gets activated, everybody's activated, but there's something kind of more in the like dynamics of how much it gets activated, or so those I was trying to think really network mechanistically about what are the classes of things that can give you that, and are there more than those two, or?
Yeah, I think so just to repeat, so the one that you said is like, for example, very, very relevant to our case, is that there are striatal projecting neurons and pontine projecting neurons in the neocortex, and there are different populations and different tasks might engage them to different degrees. So that's definitely true. The second one was that they they might talk to each other differently. And that is related to nonlinear interaction. So like a third region or neuromodulatory system might modulate the effective connectivity between different regions. Yeah, I think those are the two big classes. I don't know if you can come up with
I
another
guess there's
one.
kind of like how like the maximum firing rate in a given region when there's a task versus rest is one difference. But then there's also things like oscillation, sort of like oscillatory activity, theta and things like that they measure in other animal models.
Yeah. So yeah. So
Go ahead.
I think we could talk for the rest of the afternoon on this, and it's definitely true. Now, basically, what we're doing here is we're fitting the mean across mental activities. Like, what is the mean connectivity? And the reason why I think it is wrong. I mean, clearly that's wrong. There's clearly variations of this connectivity depending on task, and that might be informative. It's wrong having this model, but it's useful. Because if you know the mean, you can detect the violations of the mean, right? So there will be some gating, there will be some stuff. And that's really, I think, what this model is good at, is that you can say, on average, this is the answer. And now when you see deviations from it, and now that you have the mean, you can subtract out the mean, you can detect these variations and kind of detect something that might be interesting. Right? I don't think that the connectivity between cortex, the way the cortex talks to the cerebellum, is definitely going to be task dependent. It's definitely going to be modulated and definitely going to be gated. Right? It's not invariant. Right? Our model is very clearly wrong, but I think it's a useful model in this case.
Yeah.
Thank you so much. Okay, let's go to Sweden. Anders.
Hi, thank you. Thank you for this very interesting talk, Jörn. And maybe some overlap to the last question, but how I'm wondering about this sort of variability in, if you look at is it always the same parts of the cerebellum that projects to the language area or sensory area, or how big is the variation between different individuals?
Okay, so the variation is about 60%. So like 30% of the variance in the maps, or 30 to 40%, you can explain by group atlas in a group connectivity model. And the rest is small variations. Now, those small variations, these 50 to maybe 60% of variations that we see that are individually measurable, replicable in this individual, there are three sources of it. There is where these functional regions are located in the cerebellum, okay? And there's mostly small shifts. Sometimes these regions are bigger, sometimes these regions are smaller, they're slightly different. They're always kind of in the same place, but they do shift around, and our papers show that individual maps, if you want to see that. Okay, there is, of course, spatial variability in the neocortex that many people have shown, right? So, especially in prefrontal cortex, right? Like these working memory areas, sometimes they're like 3 spots, sometimes there's a banana, sometimes it's kind of like, looks like something. They're all normal subjects, but it's all arranged. So this makes it really hard to now figure out what is the spatial variability, okay, that we actually have a good analytical handle on, because we can see what can we reliably induce and what's measurement noise, what's replicable in the subject. And that comes up with this estimate of 60-40, basically. But it's very hard to distinguish that from really true variations in the pattern of connectivity. So that's actually, clinically, that's really important thing. Like, if we have an hypothesis that maybe in ADHD or so, that the way cortex and cerebellum talk to each other is disrupted or abnormal, how do we identify this given that large spatial variability? So I'm still struggling with finding a good analytical solution for that.
Thank you.
Wonderful. Sam.
Thanks, Jörn. That was awesome, as always. I had a quick question. So I was wondering, for the like unusual activity idea, could you do that also with, like, hierarchically when thinking about connectivity itself? So in other words, let's say you did a map of basal ganglia and cerebellum, and then you wanted to say, is the functional correlation I'm seeing in task A between ventral striatum and cerebellum more than the connectivity I expect, given a connectivity model from cortex to ventral striatum, right?
Yeah. I guess. I didn't sleep very well. I have a headache.
Yeah, I guess I'm just like, could you, or like you're saying like these kind of task-driven situations where the cerebellum shows greater activity than you expect from cortical activity alone. But I'm thinking, you could also do that for connectivity itself, right? Like
I
the
think so.
cerebellum.
Yeah. I think that's a really
Cool.
good suggestion. I mean, yeah, and I mean, that is a reason or motivation to collect more data sets and really building this framework. Because if you get in the second or third order questions that you're saying, like, yeah, the interactions are like mediators and so on, you really need a lot of data to do
I
this.
see. Okay.
And not just more data, not more bad data. You need like good active state data that really spans the functional breadth very well. So that really dissociates regions reliably. But I think now, like now having 450 task condition, I think we're getting into striking range of starting to do something like this. Yeah.
Cool.
So if you're
Thank you.
interested, I mean, there's the framework we are happy to make available to collaborators. So do contact us if you have really good ideas.
Awesome. Thanks,
Yeah.
Jörn.
Rich, please.
Hey, Jörn. Your last bit about the orbital frontal and the maybe connections with the olive and stuff made me wonder, have you thought about doing the above the line sort of analysis where instead of having variations on a task, levels of a task like the working memory one, do it with the tasks in terms of, or that vary in terms of the number of errors and say, look to see, do you really get the sort of above the line recruitment of the cerebellum when the errors increase?
No, but one of my graduate students just suggested the same idea to me, and I think it's a very good one. So we should definitely pursue that. Yeah, so that would be I don't know, like error-related processes. Not orbital cortex doesn't first come to mind in terms of error, general cognitive error. So you have like the anterior cingulate and so on, more there.
Right, but just in terms, is the cerebellum actually responsive to non-motor errors?
Yeah.
Or motor errors?
Yeah, it's worth looking at that. It's a really good suggestion functionally. Yeah. We've been so obsessed with heart rate over the last 2 weeks that that we haven't looked at this yet. Yeah.
Thanks.
Wonderful. Yeah, I thought the orbital frontal results were absolutely striking, and given its role in evaluating stimuli and decision making, the maximizing future reward, and its connection to the cerebellum. I don't know if you have any final words, Jörn. You want to talk about the orbital frontal cortex?
Well, let's go find it out. I mean, this is a community. I mean, if it's true, it looks very strong. So strong that I'm thinking it's got to be an artifact. That was my first gut reaction. And I think I looked at this and I'm like, I don't want to look at this for 2 months until it was so persistent that I had to take my hand away and say, like, it seems to be there. And it seems to be there in most data. It's just pretty reliable. And Ali has done things like taking out orbital frontal cortex from the connectivity model, retraining the whole connectivity model, and then see how well that predicts without and it's substantially worse, right? Especially in these areas. So you kind of need what's going on in orbital frontal cortex to explain what's going on in the cerebellum. So we've done a couple of things that already make me think like maybe it's true, but I think it's early days, and I'm not aware of any good animal work that kind of looked at. Nobody's put orbital frontal cortex, a tracer in orbital frontal cortex, and looked in the cerebellum that I know. And quantile projections are, I don't know what the data is, quite honestly. I haven't seen anything specifically on that. So
beautiful.
Yeah.
It's great to have you. Thank you everybody for coming, and wonderful stuff.
Good to see everybody. Bye-bye. Stay in touch. Bye-bye.
Bye.